So welcome everybody to this session. I think it's the 28th session of the Emerging Topics in Biomolecular Magnetic Resonance, which is organized by Lauren Andreas, Stefan Glüger, Christian Grischinger, Mei Hong, Art Palmer, Markus Zweckstetter, and myself, Oscar Millet. The idea is to present frontier research in magnetic resonance applied to biological systems, as you know. And this is sponsored by both ICMRBS and Euromar. Remember that you don't need to record the series because it will be recorded for you and then distributed and broadcasted by the ICMRBS YouTube channel. In case you had any question, you just simply type it anytime at the Q&A questions, and then it will be later on answered by the speakers. Today we have a very vibrant session with a deluxe panel, and then today will be talks by Mei Hong and Markus Zweckstetter that they will be introduced by Bob Griffin and Advax. And this is actually the last webinar for the 2020 series, and we will come next year on January 14 with also a very top series with presentations from Advax and Tatiana Polonova. With that, I think the first talk is for Mei Hong, and I leave that the panel to Bob.
Okay, so I'm supposed to introduce Mei to you. I don't think I need to do much of an introduction. She's a very well-known figure in the area of biological NMR. And she got her undergraduate degree out in Western Mass at Mount Holyoke College, and then went to California to work in the lab of Alex Pines for her PhD. She was back here at MIT for about a year as a postdoctoral student, and then went for a short stay to UMass Amherst, and then to the Iowa State University in Ames, Iowa, where she was a faculty member for a number of years. And then about 5 or 6 years ago, we enticed her to come back to MIT. Mei is pretty well known, very well known for studying membrane proteins, for example, the M2 protein of influenza A. And today she's going to tell us about a structure of the SARS-CoV-2 protein, which is a homopentameric cation channel. And she just published a very nice article on it in Nature Structural Biology in the December something issue. Okay, so Mei, the virtual floor is yours.
Thank you, Bob. So make sure this works. All right. Takes a few seconds for the slide to show. All right. Does it look fine? Okay. So great. Thank you for the introduction, Bob. I'm happy to present my lab's recent work on solving the structure of the SARS-CoV-2 envelope protein, as well as the just a little bit earlier, the influenza corresponding viral pooling. So, all right, here we go. Let's see. All right. So the RNA genome of the SARS-CoV-2 virus, which has caused about 1.6 million deaths worldwide now this year, encodes 3 membrane proteins. The spike protein, which gives rise to these large protrusions on the surface of the virus, the M protein, and then the envelope protein, E. These 3 membrane proteins orchestrate their actions at different stages of the virus life cycle. If you look at the spike protein, it is the one responsible for binding the virus to cell surface receptor, ACE2 receptor, to mediate virus entry. And once inside, the viral RNA is transcribed, replicated, transcribed, and then the virus protein synthesized. And then the M protein is responsible for assembling the viral proteins and RNA at this compartment of the cell called ergic, endoplasmic reticulum Golgi intermediate compartment. And here, the E protein's function is to make sure that the new virus buds into the lumen of ergic, so that afterwards, the new virus can be exocytosed out of the cell. So the E protein then is responsible for mediating virus budding. And if you delete the E protein from coronaviruses, then you get aberrant morphologies, you get immature virions, and such a deletion virus actually has been shown in animal models to protect against new virus challenges. So E deletion virus is potential vaccine candidate, but in addition, the E protein is also a cation channel. It conducts sodium, potassium, calcium, but the only ion that has a significant concentration difference is actually calcium between the inside of the ergic lumen and the cytoplasm. So, and this calcium conducting activity of E is associated with the cellular inflammation response. And this cation channel or calcium channel activity can be blocked by small molecules, such as this hexamethylene amiloride shown here. And this is actually a heart medication. And so this HMA is a potential antiviral drug compound for against E. So since the 2003 first outbreak of SARS, a small literature of E protein has grown. And this literature contributed significantly by Jean Torres at Nanyang Tech University in Singapore has shown that this E protein forms pentameric channels in detergent mice cells. But the exactly which residues face the pore versus lipids and the helix bundle handedness is not very clear or conclusive and seems to vary depending on what detergent mice cell you use. And meanwhile, my lab has worked on the equivalent viral poring of the influenza virus, the M2 proton channel. This is tetrameric. In lipid bilayers using solid state NMR. So since when the outbreak, the pandemic started, we turned our attention to E very quickly. So we cloned this E protein into a PET sumo construct. And because it's not clear whether any of this E protein whether the protein would express well, we tried both the full length and shorter construct, and it turns out both the full length and the transmembrane construct express pretty well. The transmembrane construct is expressed 80% into the soluble fraction and 20% in the membrane and inclusion body. And this sumo ETM fusion protein can be purified on a nickel affinity column, and then you cleave the tag with a sumo protease, and then you can purify the ETM in HPLC. So we get about 10 milligram per liter of N9, so that's pretty good yield. And we produce C13 M15 label ETM. We also prepared a fluorinated version in order to measure distances. So here in the transmembrane domain, there's 3 native phenylalanines. So we can tag these with parafluoro phenylalanine. So they're regularly spaced, and that will come up later. So even just the first look of the ETM 1D spectrum, carbon, nitrogen spectrum, show very quickly that the protein is well ordered, sensitivity is very high. The protein is largely immobilized because if we change the temperature by 40, 50 degrees, it doesn't make much difference to the intensity. And so already in the at ambient temperature in the lipid bilayer, this protein is not moving particularly. We did these experiments by reconstituting the ETM into the ergic mimetic lipid membrane with PC, PE, and also phosphatidylinositol, which is a lipid we don't use all that often, and then some phosphatidylserine, so about 20% anionic lipid with a little bit of cholesterol. I would just say this ergic membrane mixture is somewhat different from the plasma membrane composition that we're used to. If you put this, reconstitute this protein into a DMPC, DMPG model membrane, the chemical shifts are very, very similar, so it doesn't make much difference. Now, if you look at the 2D carbon-carbon and nitrogen-carbon correlation fingerprint spectra, you can see the resolution is very good, about half a ppm for carbon and better than 1 ppm for nitrogen. So we can already resolve about 2/3 of all the residues in this 31 residue transmembrane peptide. So all these bode very well for complete structure determination. And indeed, we can follow up with 3D correlation experiments, nitrogen, carbon, intra-residue as well as inter-residue correlation experiments so that we can assign all the chemical shifts. And at the end of the day, you see that the transmembrane helix region spans about residue 12 or so to about residue 34 based on the C-alpha secondary shift. Now, if you look at the C-beta chemical shift in the middle of that domain, transmembrane segment, these few secondary shifts are not in the particularly ideal helical. So keep that in mind. There might be a little bit of helix disorder. Now, knowing the backbone conformation is really just the start. for a homo-oligomeric helical bundle, you really need to get at the intermolecular distances, and you really would like to know the helix orientation as well. So all of this, right, is a quaternary structure question. And because this whole E protein is so immobilized in lipid bilayers at ambient temperature, we cannot use the rotation diffusion approach for measuring the NH dipolar bond orientation and so on. So we have to focus on the interhelical distance measurements. And here, the presence of these native phenylalanines, which we tagged with these parafluoral residues, right, is very helpful for us to measure carbon-fluorine distances. We can supplement that with carbon-nitrogen intermolecular distance measurements. And in addition, we can measure the water versus lipid exposure of the residues to sort out which residues face the pore and which residues face the lipids. And so my group has been very interested in fluorine-based long-range distance experiments. A carbon-fluorine dipolar ruler can go out to about a nanometer. And the important thing here is that we don't just want to measure one or two distances. We would like to do this quickly in a multiplex fashion by superimposing this kind of readout modules onto 2D correlation template, in this case, CC correlation experiments. And you can see how we can measure the 2D CC spectrum without the readout fluorine pulses. That's the control, and we can measure with fluorine pulses. And if you subtract, you get a difference spectrum delta S, which shows you all the carbons that are close to the fluorinated residue. So demonstrated on GB1, which has 3 tyrosine residues, which we fluorinated, we can see that these are the omega-1 dimension. These alpha carbons are the ones that are close to the fluorinated tyrosines. So we take this strategy to our ETM, where we have mixed 1-to-1 ratio the triple fluorinated peptide with a C13 labeled peptide. And we can measure these difference spectrum in a 2D fashion. These red peaks can be assigned. Now, of course, then you ask which phenylalanine is responsible for dephasing which carbon. And these phenylalanines happen to be regularly spaced, which is pretty interesting. Tells you something about organization of this viral poring. And so this assignment is important, and we can go partial way towards that assignment by correlating the fluorine chemical shift directly with carbon in this head core spectrum. And here we can resolve 2 out of 3 phenylalanine signals. This phenylalanine 23 and 26 are overlapped together. And we can make the distance measurements a bit more quantitative by measuring as a function of the read or mixing time. So we have these dephasing curves. So we take all of this information, we combine them, and we supplement the carbon-fluorine intermolecular distances by intramolecular the standard CC correlation peaks, and then the CN intermolecular correlation peaks in the mixed labeled sample. And we further supplement these semi-quantitative distance constraints with a more qualitative, but nevertheless very important water edited information, spectral information. And here the idea is we transfer water polarization to the protein, in this case, mi proton, through this kind of correlation sequence. We encode it with nitrogen-carbon correlation 2D readout. And we do this as a function of the proton transfer time, short and long. And then we divide the intensities to get a hydration map. And the bluer a peak is, the more hydrated or what water proximal the residue is, the redder it is, the less hydrated. And if you look at the hydration intensity, or the heat map intensity, you find that the hydration gradient is more or less along the bilayer normal, along the transmembrane helix, and the N terminus is the most hydrated, the C terminus is moderately hydrated, and in the vast middle you have a very dehydrated zone. This is where all the hydrophobic residues are, but here you can still see some residues are better hydrated than others. For example, this leucine 19 sticks out a little bit better. There's a valine 25 here that's better hydrated than its neighbors. And the overall hydration of this ETM in this sample, low pH ergic membrane, is quite a bit less compared to the corresponding influenza BM2 for the closed state. And so that's something to note, that this is a very dehydrated channel. And if we flip the experiment and just put our selective pulse on resonance with lipids, then we get a lipid transferred spectrum. And here you can see how these aromatic, phenylalanine residues get a lot of intensities from lipids. And so that's a complementary piece of information. So now, when we take all of these, torsion angles, intramolecular, intermolecular distances, water information, and did an explore NIH calculation of the pentamer structure, first pass did not work because the structure didn't converge. And now this reminds us that the solution NMR studies in detergent mice cells had quite a bit of ambiguity as to which residues face the pore, and the helical bundle handedness is not all that clear. And then in our structural input, we typically assume that each C13 labeled helix, let's say this particular helical wheel, could potentially have direction ambiguity to either the one clockwise or counterclockwise to the neighboring helix in terms of CF or CN distance constraints. So we generally try not to specify, and that may be an issue. And so in order to sort out this better, we modeled this 5 helix bundle somewhat by taking into account that in the middle of the transmembrane helix, the helical ideality is not great. And then we demand that our constraints, our CF and CN interhelical constraints, must be reasonable. And sometimes you can sort out which direction is the clear direction. So it's not necessarily both ways possible. And we don't want to violate any of these CF distance data. We don't want to violate water lipid data. So when we dial in all of these information, we came up with 4 additional models. The 5th model worked. And in this model, you see what we do is to put asparagine 15, this one of the few polar residues at the N-terminus, as well as the relatively well hydrated valine 25 at the same position of the helical wheel, and it's at the A position of the heptide repeat instead of D. And then we introduce a little bit of helix rotation or twist at phenylalanine 23, which is in that middle, which has some non-ideality. We rotate it by just about 50 degrees from B to F position, and then we run into no violation in terms of where those CF distances are, and we have no violation against the water lipid data. And in doing so, we also could partially disambiguate some of those interhelical distances, so we can attribute them to either counterclockwise or clockwise. So now the structure calculation works. You see, this is the 10 lowest energy structure ensemble. The ETM is a very tight and long transmembrane helical bundle, 35 angstrom long, about 10 to 14 angstrom across for the C alpha distances. The helical tilt angle is very little, very minor tilt angle, 5 to 10 degrees. More importantly, this tilt is not uniform along the whole segment. In the middle, between about residue 20 to 23, there's a rotational angle change or twist. If you look at the pore-facing residues, they're mostly hydrophobic, rich in methyl-rich leucine valine. And then as you go to the two ends, or particularly the N-terminus, you get this polar asparagine 15, whose distance to each other in this homo pentamer is short enough that we think that there's hydrogen bonding between the side chains. This glutamic acid residue is sticking out. Now, this part, we have fewer distance constraints, so you'll see more disorder. It doesn't mean that these residues are dynamic, because this whole thing is fairly rigid. Now, if you look at these 3 interesting regularly spaced phenylalanine residues, you find that they all face lipids, and they bridge, or the aromatic stacking really holds these 5 helices together. 20 and 23 face one direction, and 26 point into the other direction. So I call this sort of an aromatic hydrophobic gate that keeps this channel in this closed form at high pH. So now, just how does this viral poring of the SARS-CoV-2 virus compare with the equivalent channel proteins of influenza and HIV? The influenza BM2 or AM2, for that matter, is quite a bit more spacious in the pore diameter, and it's left-handed, so the helical bundle is left-handed. The HIV VPU is right-handed, about 20-degree tilt if you look at Opella's original literature, and it's much shorter, about 20 angstrom long. And both influenza BM2 or M2 and VPU are more dynamic than SARS-CoV-2 at high pH. And if you compare the ergic bilayer bound E with the LMPG micelle bound structural model as solved by Torres, 2 years ago, you see also quite a bit of difference. The resemblance is limited. The helical bundle is right-handed in LMPG micelles. So in the bilayer, this E protein is pretty much closed at high pH with not very special ion concentration. Now, so how do you make it open? So that's what we're exploring right now. We find that if you go to low pH without adding calcium, you see chemical shift changes at the C terminus at these residues like arginine 38 and leucine 37. And it turns out if you just assess by this water, the hydration experiment, then by increasing proton concentration going to low pH or increasing calcium concentration, then you get more hydration compared to the high pH, no calcium sample that I just told you. So we think that's how you nudge the channel to open. So this work we're now pursuing. Now, solving the structure to high resolution is really to help medicinal chemists and pharmaceutical scientists to develop better small molecule compounds than hexamethylene amiloride to plug this pore. And our investigation just by chemical shift perturbation shows that if you take your HMA to the sample, you'll find chemical shift perturbations are more or less at the N terminus very significantly, down to about residue serine 16. So the first 6 or 7 or 8, 6 or 7 residues are perturbed. And we didn't do much more than that. Then we dock HMA into this high resolution structure. And you can see at least by docking, it's the HMA might bind in a dynamic way with the hexamethylene ring pointing up or pointing down. It's not clear, or it could be across in a horizontal way. Whether experimentally it's really that case or not, well, you have to do measurements. Amantadine is another weak inhibitor of this E protein, and the chemical shift trend is very similar, but less severe shift perturbation. So HMA seems to bind better. We did this, solve the structure of E in about 5.5 months. And that's pretty quick because we just before this, we solved the influenza BM2 structure. And so that experience really taught us how to clone and purify these viral porins well, and what kind of experimental strategy would work best. So I just want to remind you that the pandemic of the 20th century, the Spanish flu, by influenza A virus had even higher fatality of 50 million people worldwide. So the amount of human suffering just is unimaginable. The corresponding viral porin is M2, and my lab and many other labs have studied M2 in great detail, but BM2 has been studied less, even though that is the flu B infection is the one that's dominant in the spring seasons of every winter. And in 2019, actually, B infection surged past A infection right from the get-go in the fall months. And if you look at the BM2 sequence and compare with AM2, it looks pretty different. Many more polar residues. The only 2 conserved residues are the histidine, which is proton selective, and then the tryptophan, which is gating residue. The proton channel activity of BM2 as assessed by electrophysiology is also a bit different. BM2 can conduct protons a little bit in a reverse direction if the inside is acidic, while AM2 is strictly an inward rectifying channel. So that's curious, and we solved the atomic resolution structure of influenza BM2. Shiva is the main author at high pH and low pH, side by side in lipid bilayers to about 1.5 angstrom resolution. And you can see how at low pH, indeed, the pore is more spacious. There's a hole here. And this structure determination, that's where we did this combined, mixed carbon fluorine labeling and distance measurements. But importantly, in the case of BM2, we got lucky that the whole tetramer assembly is dynamic in the bilayer at high temperature. So there's that uniaxial rotation, which is requisite for measuring the helix orientation. So we measure these NH bond orientations using 2DNC as the readout. We get these dipolar waves, and you can sort out at high pH, low pH. The tilt angle is somewhat different. It's about 6 degree larger tilt at low pH. Now you see at this low pH tilt angle, there's not a particular kink as far as we can tell. And so if you look at the water permeation pathway of the BM2 at high and low pH, you get this view that the pore opens more or less uniformly from, the top to bottom. This is the whole plot. And that's different from the influenza AM2 structural information that came from X-ray, almost, but to 1.0 angstrom resolution crystal structures from Bill DeGrado's lab. Here they can tell that the low pH, actually, the N terminus is a bit shut closed, tight pore, and then the C terminus is the one open, while the high pH situation is a bit in the opposite direction. So you might think of AM2 as a transporter with this way of doing the proton conduction, alternatingly open the north side and the south side. And that may be the reason that it's doing only strictly inward proton conduction, while BM2 activation at low pH seems to be just a more symmetric scissor type activation, which we think is consistent with this partial ability to conduct reverse current. Now, if you look at amino acid sequence, it turns out that there is a second histidine in BM2, and it is exactly equidistant from that aromatic tryptophan as the functional proton selective histidine. So this sequence symmetry is fully consistent with the conformational motion symmetry. We think all of this explains very well why we have this electrophysiological difference between A and B. So I hope that these two stories together show that from the technological side, now we can combine carbon, nitrogen, and fluorine solid state NMR to really get at these high resolution structures of oligomeric virus channels, and in the future, I hope to larger ion channels. And the SARS-CoV-2 helical bundle is rigid and stabilized by an aromatic hydrophobic gate of 3 phenylalanines, and the polar N terminus is responsible for small molecule binding. And I hope that the pharma companies are and working on finding better molecules to plug the pore as antivirals to complement or supplement the vaccines that are now coming on the market. The influenza BM2 channel is open in a distinct manner from AM2, and the scissor motion, which we think is enabled by the second histidine, is fully consistent and supports the bidirectional proton conduction. So this work, it takes many people in my group, but Shiva Mandela is really principally responsible for solving the BM2 structure, and then subsequently took that expertise to SARS-CoV-2, and from molecular biology to NMR to computation, and now he's busy learning electrophysiology in Rob McKinnon's lab. And Matt McKay really spearheaded the whole initial protein biochemistry effort in the darkest month of the pandemic, really heroic effort. Alex and Aurelio helped with the CF distance measurements, as well as structure calculation, and Joao now is looking at this low pH open state. And I would like to thank my collaborators and thank you for your attention.
Okay, questions for Mei from the audience, all right? There are two right here. So Fabio, it's a great talk. What is the accuracy of the carbon fluorine distances to differentiate between oligomeric states? And how do dynamics and membrane thickness change these distances?
That's a good question. The carbon fluorine distance ruler has a precision or accuracy of about 10%. As you go to 1 nanometer, the imprecision or uncertainty is 1 angstrom. And we haven't directly looked at the membrane thickness dependence of the distances, but based on ergic bilayer versus DMPC or DMPX bilayer comparison, we think that the difference is very little, because DMPC is much thinner membrane than ergic, where we use POPC and so on. So, yeah.
Okay, Cecilia has two questions. Did you try cross-linking experiments to validate the model of the pentamer? That's question number one. Mm-hmm. And number two is, did you compare the full-length E protein with the channel? I'm not sure what.
Yeah. We did not do cross-linking experiments. That's something interesting to try. Actually, outside the TM domain, the there are cysteine residues, and what exactly they're doing biochemically, structurally would be interesting to sort out. But when we have multiple cysteines, we have to be careful with the sample preparation. But yeah, that's something interesting to try in the future. We are looking at longer construct right now, and the cytoplasmic domain of E is related to the second function, the virus budding function. And so that is ongoing research. Yeah, that would be very interesting to find out just what exactly and how the cytoplasmic domain affects the transmembrane structure, if any. Yeah.
So Grace would like to know does the structure explain the mutation which destabilizes, I guess, the protein? So do you have any thoughts about that?
Whether the structure stabilize the mutation.
Not sure which mutation.
I don't know which mutation. So from the biochemical electrophysiological work, it's known that if you mutate that asparagine 15 and that valine 25, you abolish the channel conductivity. So that is a mutation that these are the mutations that are known, and I don't know what other mutations that would be interesting to look at. I think our attention would be first on the wild type protein and really understand it, everything about it as much as we can. Yeah, probably.
So an anonymous attendee wants to know: Are you set to explore interactions between SARS-CoV-2 E protein and the MS proteins, M/S proteins?
Yeah, we would love to. This would be very interesting. And absolutely, yeah, that's, am I set? I mean, there's so many things to do, yeah, it'll be great to work on this. Yeah, I mean, there are only 3, and I think the S to E interaction would be the more interesting and probably more relevant one to target, yes.
And Rudra wants to know, considering the exactly sequence similarity of the TM domain in SARS-CoV and SARS-CoV-2, can the 2 different structural models partially be attributed to different membrane mimetics?
Oh, yeah, we used identical sequences what the Jean Torres lab used. And so I think the different structural models really come from, it's a combination, most likely, of the membrane mimic and the structural constraints, how many constraints you have, all right? So the true structure in detergent micelles, what it is, you will need a lot of constraints. And the solution MR studies, one study had a lot more interhelical distance, but no RDCs. And then there's another study that had more RDC, but very few interhelical distance. So it's like part of it is just they didn't have enough constraints. But then in addition, there's possibly true structure differences between the detergent and bilayer. Yeah.
And Oleg wants to know if you tried multiple quantum F19 NMR in this system, and the interhelix F19, F9 distances are short enough so that you might see these transitions.
Yeah, that's a good point. That's a good point. So yes, if we just put a single fluorine labeled residue per helix, then we will be able to use multiple quantum or fluorine codex kind of methods to pinpoint, to first verify that we have a pentamer, but more importantly, to try to get more precise distances. Yeah, absolutely. That's a good idea.
So Yubing wants to know if you tried to fit the NMR data to other oligomeric states, for example, the hexamer.
Yeah. We didn't have time to explore other oligomer states. And this, we really lean on the previous solution NMR data and gel data, not just solution NMR. I mean, the gel data to say that this is a pentamer, they see bands, and you have multiple bands, you go up to pentamer. So so this is, again, I mean, it's a quick study. We can sort out everything in 5 months. So yeah,
So Reno Esposito wants to know how did you incorporate F19 for phenylalanine in the expressed peptide?
So we did something, it's a fairly simple method known in the literature. We fed glyphosate right before IP2 protein expression. And glyphosate stops the aromatic biosynthetic pathways in bacteria so that you can feed fluorinated aromatic residues in there. And that's all. And that this method, glyphosate, it only compromises the yield a little bit, 20%, 10%, not too bad. Yeah, so that's all.
So Sang-Ho Park has a question. Any possibility that the rotational diffusion of the E protein is restricted by sample conditions, such as high protein to lipid ratio? Yeah. And also, did you have experimental data showing the hydrogen bonds between the N15 side chains?
Yeah, these are good questions. Sang-Ho, I was really hoping that you would have measured the rotational, the orientation back in June when you gave your talk. Because that would really help to, nail the structure better. But I don't think it's the protein to lipid ratio, because we use similar kind of, ratio for M2, and no problem, it moves. And with 1 to 20 or so, you still have a lipid excess. That's something like 4 or 5 times. So that's enough to let, the support motion, if the motion is to happen. So I don't think that's the reason. And in terms of side chain hydrogen bond, we don't have direct measurements to show hydrogen bond. So this comes from, the structure calculation that says all of these N15s, asparagine 15s, are close enough to each other. So yeah.
And Carol Post wants to know if you could describe in more detail how the structure can rationalize unidirectional transport for the influenza channel.
Yeah,
so the M2 rather than
right.
Yeah.
Right. So that's a good question. The AM2 activates by, well, at the high pH, the north side, the N-terminal side is more spacious. The C-terminal side is shut, very close. The histidine is neutral. You go to low pH, the C-terminal side is wide open. All the histidines are having electrostatic repulsion. The M-terminus, as seen at least in the crystal structure, is not actually all that open. So in that sense, it is like a transporter, alternating access, open on the north and open on the south. And then the protons would then, well, there is a concentration gradient. Outside is low pH in the normal situation. So then the protons will go only one way. And okay, what prevents, and so you say, all right, well, maybe the virus interior becomes acidic. What prevents the protons from going backwards? Well, that's where the tryptophan gate is doing. That residue blocks the proton. And when we mutated, my group mutated that tryptophan to a phenylalanine, then we find spectroscopically the protons from the interior could then access the histidine from the bottom. So it's all that tryptophan that's doing the one-way valve. It's serving the role of a one-way valve, yeah.
And finally, Cecilia has one more question, is did you try any mutants yet to look at mutants?
Not yet.
No. That's probably on the agenda. would be a nice thing to put on the agenda, right?
Yeah.
Okay. Okay, so that's all the questions. So listen, Mei, thank you very much for a beautiful talk on what is currently a very interesting and hot and concerning system for everybody in the world these days. Okay, so I hand over the floor to the next introducer, I suppose, which is Ad Bax. And Ad wants everybody to tell him what his background is on his his picture right there. Does anybody recognize it? Ad? We can't hear you there.
Okay, good. So this should work. I'm not allowed to tell you. I would probably get fired if I tell you what the background is, because people might perceive it as being a political background. It's a pleasure to introduce the second speaker this morning, who also doesn't really need an introduction as a co-organizer of this series. But I'll go ahead anyway. Markus Zweckstetter currently heads the senior research group at the structural biology in dementia.
I didn't quite catch that. Could you please repeat it?
Sorry, my phone is giving me grief here. Turn it off. He heads the senior research group Structural Biology and Dementia at the German Center for Neurodegenerative Diseases. He's also a professor at the medical school in Göttingen, and he leads a research group at the Max Planck Institute in Göttingen. Markus got his undergraduate degree in physics in 1996, and he got his PhD at the Max Planck in Martinsried. He's of course very well known for all his his prowess in magnetic resonance spectroscopy, particularly biomolecular NMR, Mars, membrane proteins, tau, and whatever have you. But he also was a postdoc in my group, and I figured I'll give him a little bit of a hard time over this. He was a memorable postdoc for several reasons. Not only was he very prolific, as many of the other postdocs that have come through my group, but he was the first one to beat me at my own game when we went for lunch runs through Rock Creek Park. Just once I was actually able to beat him, and that was on a day that it was 37 degrees centigrade. And on the way back, he got a heat stroke, and he ended up puking in the bushes and showed up about a half hour later looking very white. So the other memorable fact from the days back at NIH that everybody else in the lab remembers him for was that he was the first postdoc to beat not just Attila Szabo, but also Bill Eaton at their favorite game of tennis. So yes, you left a long-lasting memory, Markus, and we're looking forward to your scientific presentation.
Thank you, Ed. Yeah, I hope I'm not only remembered for sporting activities. Yeah, thank you. So I share my screen. Okay, so the idea of today's webinar really was to also present some of the work on SARS-CoV-2, which is done by the organizers. And Mei Hong already gave an excellent introduction and beautiful data into this topic. So my group was interested not so much in the membrane proteins, as we'll see, but more in line or along a research line, which we have followed now for a number of years, which is biomolecular condensation. So how did we come to this? And again, this basically goes back many, many years. So in the beginning of the 2000s, when we started to work on intrinsically disordered proteins. And as you all know, intrinsically disordered proteins are very nice to NMR, or NMR is very nice to intrinsically disordered proteins. And what's, I think, also very nice, which probably in the beginning of 2000s one wouldn't have expected, is that at least in 2020 now, even biologists know that intrinsically disordered proteins have to be described by an ensemble of conformations, which I heard today. So it sort of made its round. So what we now want to use in the future, of course, is to use the 1.2 gigahertz, which is here on the right, and which has been installed in Göttingen to study these proteins. Now, over the years, we really have used NMR spectroscopy to provide an atomic resolution view of these ensembles for a number of, from our perspective, important proteins such as alpha-synuclein, tau, also parts of tau, and some other proteins as you see here. And over the years, we used basically, or we refined our approaches also in collaboration with a number of other groups, using various NMR parameters from RTCs to NOEs to PREs, and also, for example, what you see here on the left-hand side, for example, for this serine arginate region of the SRSF1 proteins, where we combine a lot of NMR parameters, including J couplings, which I guess Ed will like, with molecular dynamic simulations. In addition, we, for example, also determined the structure of a complex here within intrinsically disordered regions, which fold up on binding. And more recently, which will become important later, we also have begun studies on the C-terminal domain of the RNA polymerase II. But I want to start just with a little bit of look back, let's say, on this SR region, so serine arginine-rich region of SRSF1. So as the name already says, this SR region, which follows two RRM domains, is highly rich in arginine and serine, as you can see here from the sequence. Now, using a variety of NMR parameters and molecular dynamic simulations, we found that the ensemble is very diverse and very flexible in the non-phosphorylated state of this SR region. However, it gets more rigid and more defined, is again derived from a combination of molecular dynamic simulations and NMR once it's phosphorylated. And this is very clear, for example, if you look at the heteronuclein measurement of the peptide here, either in the non-phosphorylated state or in the phosphorylated state, you see that the het NOE basically increases from about 0 to about 0.4, 0.5, really demonstrating a drastic dynamic switch upon phosphorylation in this SR-rich region. And just maybe you can keep this in mind, that's why I highlighted it in black. The kinase which we used here for phosphorylating the SR region is SRPK1, which is extremely specific for phosphorylation of SR motifs. Now, let's jump to SARS-CoV-2, and I don't have to give you an introduction to the biology here. I just want to point out, Mei spoke about the 3 membrane proteins, and the 4th structural protein of the virus is the nucleocapsid protein, which one of its function is to wrap around the viral RNA and thereby protect it from the host cell environment. The nucleocapsid protein is also very interesting because it's expressed at high levels in infected cells. It has been shown to be important for viral assembly. And again, something which will become important later, it has been shown to be involved in RNA transcription and viral replication. And because there were some comments made before about antibodies generated in patients upon SARS-CoV-2 infection, this was also tested. And what you see here now, very early study actually from patients from different institutions here, including NIH actually. And so there's 100% sensitivity for nucleocapsid when you sort of look at patients 14 days after infection. That's even more specific than for the spike-2 protein. So another important sort of aspect to keep in mind, and of course, I mean, the SARS-CoV-2 is not the first one. We've heard about SARS-CoV and many other viruses. So we can learn a lot about looking back at what has been found of these previously studied viruses. And one important observation there was for negative strand viruses in this case, that this viral factories, so what we have here, right, that the nucleocapsid protein is involved in RNA transcription, viral replication. This viral replication machineries have been shown to have liquid-like properties. For example, here in this microscopy images, you see that these two dots, so to speak, fuse a characteristic feature of phase separation and liquid-like properties. And this was actually this year also studied by Martin Blackledge, and he showed that the combination of the nucleocapsid protein and another protein called phosphoprotein can actually undergo liquid-liquid phase separation and promote nucleocapsid assembly. In addition, actually, in addition to the nucleocapsid and the phosphoprotein, there's also the requirement for the L protein in cells. So we were wondering then if that's SARS-CoV-2, which has been shown to be important for replication, might also be involved in a phase separation process, and this might be important for viral replication. So let's have a look at the structure of the N protein. So the domain organization is shown on top here. So it contains a lot of intrinsically disordered regions. So at the tails and also the linker region between an RNA binding domain and a dimerization domain. And a lot of structural work has been already done there. So actually very recently, the crystal structures of the RNA binding domain and the dimerization domain have been published. There's also been done a study where people looked at the binding of RNA to the RNA binding domain using NMR. And of course, there's the huge effort by the COVID-19 NMR consortium having already assigned some parts of the protein. We were interested in the SR-rich motif, coming really from our previous studies on the SR region of SRSF1. So we wanted to focus on this part of the protein. Before going that, we were investigating, motivated by these previous studies, that the viral replication machineries in these viruses can be liquid-like. We investigated if the nucleocapsid protein of SARS-CoV-2 can undergo phase separation in the presence of RNA. And this is indeed the case. So it very nicely forms this droplet-like structures under the microscope, and we can determine the phase diagram and find that phase separation is strongest or is promoted by charge neutralization between the nucleocapsid protein, which is predominantly positively charged, and the negatively charged RNA. At that point, I should point out, because of course, I mean, this is a very, let's say, intensively researched area currently, that independent studies from the labs of Nick Fasi, Alex Hollas, and Amit Ledfelter also showed that the nuclear capsid protein of SARS-CoV-2 can actually undergo phase separation. But we wanted to understand this a little bit further using NMR and molecular dynamics simulations. Before going to that, just to remind you that what is the connection of now this in vitro observed liquid-liquid phase separation of the nuclear capsid protein with RNA to a potential toxic effect, in the sense an effect which modulates the host cell response. And a number of studies has shown that various viruses actually interact and inhibit or divert RNA containing stress granule assemblies. So stress granules are easily formed in the cell. So if the cell experiences stress, the RNA falls off and binds to various RNA binding proteins, and the stress granules are formed. And so the viruses, so this is a host defense reaction, and the virus try to modulate this host defense reaction to be more effective. So for this reason, we then evaluated if the nuclear capsid protein of SARS-CoV-2 actually can also colocalize to stress granules, and this is shown here. So G3BB1 is the canonical stress granule marker, as indicated here, and the N-SARS-CoV-2 protein nicely colocalizes into this or with the stress granules when the cells are stressed, indicating that the N-SARS-CoV-2 or the SARS-CoV-2 virus might also modulate the stress granule response of the host cell. Now, how about the SR region, right? I introduced this by using the SRPK1 protein and our ensemble determination there. Why did we believe that this is why it's particularly important to look at this region? And this is indicated here. So there has been some proteomic studies been done and post-translation analysis studies on a cell-wide level. And it was shown that the SR region, which is indicated in here, is actually the most highly phosphorylated region of the whole nuclear capsid protein. In addition, this is an analysis actually which aligned it during this study. When we look at the mutations, and at that time, which was I think in June or so, we pulled down more than 40,000 sequences from the deposited genomes of the SARS-CoV-2 virus. And again, one finds that the SR region is actually the most heavily mutated region in the nuclear capsid protein of SARS-CoV-2. So that, of course, motivated us to look at this region. Another actually region, and this is a very recent one, it was put on bioRxiv, is that when one uses an inhibitor, SRPIN 340 against SRPK1. So just to remind you, this is this kinase which specifically phosphorylates SR motifs in SR-rich regions, right? Then this basically completely knocks out the infection in SARS-CoV infected cells. So the reduction in SARS-CoV-2 infection titer is decreased by more than 5 orders of magnitude. Again, highlighting that this region might be really critical for infection and replication of the virus. So how does this region actually look like? So here's some amino acid sequence, and we actually focused on the region from 182 to 197 because that is the region with the largest amount of arginine and serine residues. It actually has 9 serine residues and 4 arginine residues. We then did some NMR measurements here. So what you see here is a natural abundance protonitron HSQC of this SR region of the nuclear capsid protein. And the spectrum looks beautiful. We assigned it using traditional methods, and then did a secondary chemical shift analysis using Talos Plus developed in ETSLab. And what we found is that, as you see here in the green line, mostly this is disordered, this peptide, and there's very little alpha helical or beta strand conformation. However, we then actually zoomed in a little bit in this blue curve, right? Because as you see, close to the arginines, it seems that it always has a sort of a little bit of a hump. So if you zoom into this region, and again, plot here the chemical shift derived secondary structure, then you see that at around this arginine, which is arginine 198, there's a peak in the alpha helical propensity for this peptide. And to support this, we then performed molecular dynamic simulations using state-of-the-art force fields. And again, we see that there's a tendency for alpha helical formation in proximity to this arginine 189. Our motivation for doing that, of course, was to understand how RNA induces phase separation of N-SARS-CoV-2 and how this might be regulated by mutation and phosphorylation of the SR region. The next step, we used then mass spectroscopy to better understand the binding of RNA to the SR region. And as you can see here, a two-dimensional proto-proton correlation spectrum. When we add the poly-U, we see continuous chemical shift changes, in particular here for the arginine residues, which are marked here. And I want to point out again, this arginine 189, which I showed you before, has this helical conformation because that shows very pronounced chemical shift changes. We then quantified that, and this is the blue curve. And as I just showed you in the spectrum, the arginine 189 has the most pronounced chemical shift change. And then we compared it to MD simulations of 5 SR peptides in the presence of poly-U and the number of peptide RNA contacts. And what we found is that actually the arginine 189 makes most of the contacts with the RNA. And what is really striking, at least was striking in my eyes at that time, is the gray curve, because the gray curve is again the mutation frequency seen in more than 40,000 SARS-CoV-2 nucleocapsid sequences, right? So many of these residues have been mutated, while the arginine 189 actually has been spared, suggesting that it's really functionally important. So why is it exactly the arginine 189? We don't really know, but one hypothesis would be because it's exactly this arginine 189 is this in this transient alpha helical conformation, so that the structuring in the peptide helps binding to the RNA. So our next question is, what is the impact of SR phosphorylation? SR phosphorylation, I pointed out that this is very important as indicated here, and is widely present in the cell, in the infected cell. So the first thing what we did is we looked for protein phase separation, in this case using turbidity. So that's the turbidity what you see here. If there's no phase separation, you would expect a turbidity roughly of 0, of course. And when you get phase separation, the sample, if you look at it, gets turbid and you can measure this. And the blue curve is for the non-phosphorylated protein. And the red one is now for the phosphorylated protein and phosphorylated again by, let's say, favorite kinase SRPK1. And as you can see, the phase diagram shifts when the nucleocapsid protein has been phosphorylated by SRPK1. So why is this the case? Again, we used NMR for this. So with the titration now of the unmodified as well as the phosphorylated at SR region with poly-U again. And as shown before, the poly-U nicely binds to the SR peptide if it's not modified. But if we have one phosphorylation and the phosphate group is actually serine 188, as NMR can show, or if you have two phosphorylation sites or two sites phosphorylated, then we either largely inhibit or we completely block binding of RNA to the SR region. So this could be, of course, because of electrostatic considerations, but we wanted to understand this a little bit further. So we did, again, molecular dynamic simulations and investigated intramolecular as well as intermolecular contacts in these peptides. And for peptides with increasing degrees of phosphorylation, as you see here on the right-hand side. And I show this in the bottom right away. So we see that the number of intramolecular contacts increases with phosphorylation, right? It saturates at around 3 to 4 sites of phosphorylation. So when you have charge neutralization, and this is also represented here. Now, what I want to point out is if you plot this in a sequence-wise, a specific manner, that it's particular the arginine and the serine, which basically point to the same direction or to the same side of the chain, right? These seem to be making predominant intramolecular interactions, where you see less of the intramolecular interactions if the serine and the arginine point on opposite sides of the chain. So again, basically represented here, if the serine and arginine is on the same side of the chain, we have the contacts. So you see a strong increase in intermolecular contacts and stabilization, and this is really highly consistent with our previous studies of phosphorylation of the SR region of SRS1. Now, why does RNA binding decrease? And this is also illustrated here. So for that, we analyzed the intramolecular contacts, both between different SR peptides as well as between SR and RNA. And similar to the intramolecular contacts, the peptide-peptide contacts increase with phosphorylation simply because you have salt bridges between arginines and the phosphorylated serines, while at the same time, in a phosphorylation-dependent manner, the protein or peptide RNA contacts decrease, providing a rationale, first of all, why we see by NMR really an inhibited binding of RNA to the phosphorylated SR region, and also an explanation for the observation that phosphorylation modulates the phase separation of the nucleocapsid protein. So this brings me back now to this viral replication factories and phase transitions. So the transcription, or the major enzyme for transcription of the SARS-CoV-2 is RNA-dependent RNA polymerase, and the lab of Patu Kramer has solved the beautiful crystal structure of NSP12, which is actually the polymerase, plus two helper proteins NSP7 and NSP8. So if now our phase-separated compartments provide, let's say, in simplified in vitro system for a virus replication compartment, we were expecting or hypothesizing that the RNA-dependent RNA polymerase will concentrate into our nucleocapsid RNA droplets. And this is what we found. This was prepared, the protein. Actually, we got this from Patu Kramer's lab. And then we added it to our assay, phase separation assay, and we see that the nucleocapsid protein concentrates into the droplets together with the RNA-dependent RNA polymerase. And the whole process is very specific because it depends, again, on the phosphorylation of the nucleocapsid protein by SRPK1, so the phosphorylation in the SR region. And this attenuates both the recruitment of the full RNA RDP complex as well as of just the polymerase itself, indicating that the SR phosphorylation of the nucleocapsid protein can really regulate this. And this brings me now basically to our previous study, where we looked at the phase separation and clustering of the RNA polymerase II, driven by the C-terminal domain. And because we showed previously that this C-terminal domain of the RNA polymerase II, which is intrinsically disordered, highly repetitive, phase separates. But moreover, we showed in collaboration with the people in Berkeley that the RNA polymerase II clusters in the nucleus of eukaryotic cells. And this clustering depends on the length of the CTD domain, of this intrinsically disordered domain, which suggested to us the following model is illustrated here, that at the beginning of initiation of transcription, the RNA polymerase II clusters here, and this clustering is driven by the C-terminal domain and held by transactivation domains. And once it's phosphorylated, then the polymerase is released from the cluster and actually can go into elongation. So if you take now this into together with studies which we found for SARS-CoV-2, right, where we saw that we get the concentration of the nucleocapsid protein and the RNA into these droplets, and into this then highly condensed protein nucleic acid dense compartment, we get further recruitment of the RNA polymerase. This really suggests that what we have here is an in vitro reconstituted viral replication compartment. So together, this showed that the studies showed that the nucleocapsid protein of SARS-CoV-2 can undergo RNA-induced LPS. What I didn't have time to show is we also see that the droplets change their properties in a time-dependent manner, suggesting that this might be the early stages of nucleocapsid assembly. We find that the protein can associate with stress granules and such thereby modify the host cell response. Phosphorylation is a critical effect, right? And I showed you that when you use an inhibitor against SRPK1, you can reduce the infectivity by 5 orders of magnitude. And very importantly, this might really be related to the formation of this nucleocapsid-driven viral replication machinery. And so, of course, we want to further gain insight into these questions. And for that, we want to use the 1.2 gigahertz spectrometer. And the first spectrum, which we got from this machine, which, as you can see here, if you read the date, it was today. And Milan actually finished the spectrum probably an hour ago or so, a little bit than an hour ago, because the spectrometer was just recently sort of given to us and is kind of still now stable on field. But what you see here is now basically the methyl proton region to the correlation spectrum of the full length nuclear capsid protein at the 1.2 gigahertz. And maybe you look at this and you say, yeah, I mean, that looks okay, but it's not fantastic. I just would like to remind you that on first of all, this is a 90 kilodalton protein, which was unlabeled. So this is at natural abundance at a protein concentration of 50 micromolar. So we expect with further sample improvement and so on to really get nice methyl trosis spectra, which we then can use to investigate the interaction with the viral replication machinery. This brings me to the end, and really the most important thing, I really want to thank the three people here, Adriana Savastano, Alain Ibanez de Apacua, and Maria Rankovic, which really took on the project in April, I think, this year. And in a very concerted and not only concerted, but extremely collaborative effort, pushed this project through. And more recently, Milan Zikratla joined the project and will be working more on the NMR part. The proteins were given past sometimes by Patikrama. And all this would not be possible really with the full support of the lab. And I would like to thank you for your attention. Thank you.
Thanks very much, Markus, for a very inspiring talk here. This was fantastic. Let's start with some questions. First, there was an anonymous question. Let me try to find it back. No, let me start from the top here. Hashim says, great talk. How does magnesium affect RNA protein binding and phase separation? Do you see similar binding and phase separation with a stable RNA duplex instead of poly-U? Does RNA 2Y structure play a role?
Mm-hmm. Yeah, a very good question, Hashim, of course. Yeah, in a way I would have to answer like Mei did. Of course, there was always extreme time pressure. And my three people could only work 24 hours a day for a limited amount of months. So that limited also a little bit the number of experiments which you could do. But so for the magnesium, I don't know. I expect, that's a very good question, of course. With the RNA, I also expect, and actually this is an aspect which is particularly looked at by Ami Glattfelter, right? Because the viral RNA has very specific properties sort of along its sequence. So there will be some influence by the secondary structure of the RNA. At least that's what the studies from Ami Glattfelter suggest. But we haven't really looked at, for example, we haven't used poly-A or so, but I mean, this is part of the things which we will look at. Yes, thank you, Hashim.
Great. Rino Esposito says, congratulations for the nice talk. I seem to have seen that the spectrum of your SR-rich region displays major dispersion when you add two phosphate groups. Is this correct? And maybe I can follow up. How specific is the phosphorylation? Do you know? Is it sort of like random, or
okay, so
are there preferred targeted sites?
Yeah. Yeah, I think that's, for example, definitely a strength of NMR. So maybe I didn't show so many NMR spectra, but we really tried very hard to identify the phosphosites in the SR region by mass spec. And because of the highly degenerative nature of the sequence, we failed multiple times actually. Whereas with the NMR, and of course, you know that especially serine and threonine phosphorylation is very nice in intrinsic disorder protein because it shifts to a unique region of the spectrum. we can both identify not only the sites of phosphorylation, but also quantify it. In this case, actually, we found that the site which is first phosphorylated by SRPK1 is serine 188, which is exactly the serine before the arginine 189, which sort of is the key residue for the interaction with RNA. Even more independent studies have suggested that maybe this S188 is indeed a key phosphorylation site, in the sense that it also primes phosphorylation by other kinases, because in some of the proteomic studies, all the other kinases have been identified, but from some experiments which we already have done, we see that in the tests which we've done, that really you need first SRPK1 to phosphorylate S188 to get phosphorylation by other kinases in a quantitative manner. So it's very specific, yeah.
Thanks, very interesting indeed. A question by Shenki Chang says, does aspartate or glutamate substitution have the same effects as phosphorylation of serine on the liquid-liquid phase separation?
Yeah.
Have you guys tried?
I mean, we haven't tried it because, I mean, this is complex coacervation, meaning driven by electrostatic interaction, at least in our case. And of course, I mean, a glutamate introduces one negative charge or so it will partially mimic this, but probably not fully. So I think in that case where we know, or where we at least have very good candidates for kinases which are relevant, we prefer to use the kinases actually. Yeah.
All right, makes sense. Yuho Lee is asking whether it's possible to estimate the number of proteins or molecules in your droplets. Perhaps from diffusion or from some other kind of...
Yeah, I mean, one could do a very rough estimate from fluorescent microscopy. But actually, I mean, I don't have time to go into this, but actually NMR is the best method to do that. One cannot directly do it in droplets, but you can sort of let the sample settle down and make a condensate, as many other people which do NMR on these systems have done. And then actually using NMR tools, you really can very accurately quantify or determine concentrations inside and outside, and how this changes with different factors. So I think that's NMR is probably the most accurate tool for these studies. Yeah.
All right. Dorothy Kern is asking whether you can differentiate if the charge repulsion between the phosphate groups in the RNA or the changes in the intramolecular interaction within the peptide are the main contributor to modulate the peptide RNA interaction. It's a little bit of a chicken and an egg question, I guess. But
I mean, what we see, right? I mean, I maybe rephrase it in the sense of, let's say, unspecific electrostatic interactions versus, let's say, specific intramolecular contacts. I would favor intramolecular contacts, right? Because as what I showed you for the SRSF1 protein, I mean, we see really a very strong rigidification by the NMR dynamics measurement upon phosphorylation of the peptide, which really suggests that you have specific intramolecular contacts at least. I mean, the intermolecular ones we just can deduce from the MD so far, but that I would really favor in the sense of specific salt bridge formation between actually an arginine and a serine, which point on the same side of the extended chain. Yeah, as shown by the MD.
Sorry, related to this, do you think that measuring the interaction at different phosphate ionic strength would help?
Yeah, that's also a good suggestion.
Because perhaps you can rule out the non-specific electrostatic interactions just by simply increasing the amount of phosphate.
Yeah, I think there's definitely a contribution, at least for phase separation. There's definitely a contribution of, let's say, non-specific electrostatic interactions, because we see that we get maximum phase separation upon charge neutralization, so matching, let's say, the charges of the protein in the RNA. Yeah.
All right. Another interesting question here from Martin Blackledge. He says, great talk, but that's usually the introduction to a difficult question. There's a lot of evidence that RNA replication occurs predominantly in double membrane vesicles in coronaviruses. Do you think that RTC-associated droplets may play a related role in replication, trafficking, for example?
Yeah, I could, I mean, of course, I will now say I could easily imagine, but I will also back this up by saying that that in SARS-CoV, for example, it has been shown that the nucleocapsid protein can interact with the M protein. And we've heard about the M protein from Mei already, and that it's part, or that it's important for the vesicle budding and so on in the ERIC. One hypothesis would be that actually you get phase separation driven by the nucleocapsid protein on these membranes through the interaction of the nucleocapsid protein with the M protein. But of course, this is pure speculation, but it's based at least on the fact that the two proteins can interact in SARS-CoV. Also, it hasn't been shown, I think, in SARS-CoV-2. Yeah.
Very interesting indeed. Actually, my own question is probably a very naive biological question, which may surprise you. It is known that the infectivity of viruses goes way down further from the time of infection. So after 5 days, people have high infectivity per variant that is produced, whereas after 2 weeks, you're still very rich in virus production. So the viral load in saliva or sputum is very high, but the infectivity goes way down. So is it conceivable that this is related to kinase activity of the host system, that is the patient when it's under pressure from the disease? Or is there a possibility that it could have a modulatory role?
Yeah, I mean, so I mean, I'm also not a biologist, as you know, but I guess it could be. I mean, for example, in this phospho maps, which have been derived from infected cells, and I think in different infected cells now, there's constantly kinases or phosphorylation, and therefore kinases being identified as playing a role. I mean, this also has been suggested that the kinases are really a drug target. Also related potentially to the stress granules and so on. As far as I'm concerned, this one study which I showed, which is now deposited in the bioarchive, this is where they really tested this with inhibitors. And as you've seen, I mean, that the effect is, as far as I can tell, dramatic, right? You increase the infectious titer by 5 orders of magnitude by simply inhibiting this one kinase, right? Actually, it's if you look at the paper, they say it's the two kinases because the inhibitor itself is not only inhibiting SRPK1, but also the isoform SRPK2. But the major one seems to be SRPK1 because they did some further experiments. So it seems to be extremely specific, actually.
Yeah, very intriguing indeed. Question from Fabio Almeida. How does the assignment of small peptide of the SR region match the assignment of Roberta of these regions presented in the COVID-19 NMR consortium? And following up on that, I was disappointed to see no assignments on that spectrum you recorded on the 1.2 gigahertz this morning.
Yes, okay.
Now, a couple of hours later.
I will tell Milan that you have to work harder. I know this from you, right? Coming in the morning and in the evening. Yeah, so I mean, we did compare some of them. We also, which I didn't have time to show, we also have HSQCs, nitrogen HSQCs of the full-length protein. And but again, these are very fresh. And we compared it both to, I mean, we didn't have Roberta's assignment, but we of course compared it to the assignments from the COVID-19 NMR consortium. And actually, the assignments of the folded domains basically perfectly fit, despite the fact that we had slightly different buffer conditions. And for the intrinsic disordered regions, yeah, it depends a little bit on which intrinsically disordered region you look like. For the SR region, there's some fit, but not full fit. And this is likely because there's been a hypothesis in the literature that the SR region kind of folds back on the RNA binding domain in a kind of maybe auto-inhibited state or something like that. Yeah, but so this is ongoing actually, Fabio. Thank you.
All right, a question from Gue Yin, whether phosphorylation regulates the phase separation, or he starts by saying, he or she, hard to tell from the name, phosphorylation regulates phase separation of the srRNA. Is there such possibility that during this phosphorylation process, the kinase could be recruited into the condensed phase?
Yeah, that's a very interesting research question, because, right, I mean, these condensates are believed to be, or some condensates are believed to be actively regulated. And of course, this would involve potentially ATP and so on, and the recruitment of the kinases to the droplets. We've seen this for other droplets, such as tau, where we see that the kinase goes inside the droplets. We haven't measured this for the SARS-CoV-2 droplets, but again, good point, yeah.
Yeah, interesting indeed. Dorothy Kern has another question, whether you know which human phosphatases are reversing the phosphorylation. Is that known, or do you know? Or
that's a fantastic question, Dorothy. I don't think, I mean, people have looked at yeah, which kinases and so on by proteomic studies. Maybe they also found some phosphatases, but I cannot tell now. And I'm not aware that, for example, with inhibitors or something, people already tried to target specifically phosphatases and looked in infected human cells or monkey cells or so. Yeah.
One more interesting question here, whether you know perhaps whether from Madhan Kumar Mohan, whether you know whether the RNA contact perhaps particularly involves the side chain of the guanidinium group of arginine 189, considering that serine 188 is so critical.
Yeah, that we definitely should measure. And of course, this is easy to do by looking at the proton nitrogen HSQC of the guanidinium group. Because, I mean, that's, for example, we did again for tau with phosphorylation. There one nicely sees, so in the unphosphorylated state, right, the side chain signals of the guanidinium group are kind of weak because of the rapid solvent exchange and cluster in one region. And when you get a formation of a salt bridge, it really shifts out the peak in a unique position. So I think with the S81 phosphorylated, which we can show it is, and the small peptide using nositoxy, we can definitely identify whether it's making the salt bridge. Most likely this is the case, because that's again what we've seen in tau, that these intramolecular salt bridges induced by phosphorylation tend to be to the nearest neighbor. So let's say to residue I or to residue I -1. We didn't see this so far for, let's say, more long range, which then would lead, let's say, to a kink of the chain. So far, this was always, let's say, nearest neighbor, or almost nearest neighbor at least.
Yeah. Question from Grace Royapa, whether you speculate whether this region will bind better in the presence of the RRM domain. Yeah,
so actually we know that this is the case, the data which I didn't show you, and maybe the question is still in the Q&A, because what I didn't show you, how the phosphorylation affects the recruitment to the stress granules. Of course, we did that also. And what we find there is that the nucleocapsid protein, which has been phosphorylated by SRPK1 at this S188 and actually 3 or 4 other sites, it still can co-localize to stress granules, also maybe a little bit weaker. We didn't properly quantify that, but if one looks at the microscope image, it looks at the recruitment is a little bit later, a little bit less. but that's actually expected, right? Because the RRM domain will bind RNA. And as I introduced, stress granules are full of RNA. So it's probably a cooperative process between the RRM and the SR region. And this will be regulated then by phosphorylation through different kinases, SRPK1. But as I said, SRPK1 is a priming kinase. And then there are some other kinases coming in, which might also phosphorylate the RRM domain.
Makes sense, yeah. Last question here by an anonymous attendee, always very scary. Markus, on slide 3 of your presentation, what experimental restraints were used for the structure of synuclein? Why does the structure look like a donut?
Yeah, yeah. This, I mean, of course, this is an ensemble, and I tried to use that for illustration, which is always not a good idea, of course, for intrinsically disordered proteins. So at that time, actually, I mean, this we really used, I mean, like I think Explore NH structure calculations at that time where we used PREs and the hydral angles and so on. I think in the meantime, I mean, other people, not we, have developed better approaches, of course, to calculate structural ensembles, in particular Martin Blackley's group, Julie Fagan, but also Gerald Hummer, for example, maybe in collaboration with Ed and maybe a little bit now with us also. So I think there are better approaches for calculating ensembles. So the donut is maybe making you hungry if you didn't have breakfast yet on the West Coast, but other than that, I think it doesn't mean anything.
Great. I think this sort of wraps it up. This was a fantastic set of seminars, great questions by the audience, and I think it's a fantastic end of the year for NMR, although not necessarily for the planet as far as the virus is concerned. Back to the organizers.